Welcome to the Molecular Therapy Podcast. I'm Dr. Rory Bricker Anthony, Scientific Editor of the Molecular Therapy family of journals. In honor of Molecular Therapy's 25th anniversary, this episode is the first in a series hosted by Dr. Joseph Glorioso, Editor-in-Chief of Molecular Therapy, that will cover comprehensive reviews of critical developments in the field of gene and cell therapy over the past 25 years by expert authors. In this episode, Dr. Glorioso will discuss two of these reviews: Gene Therapy Then and Now, a look back at changes in the field over the past 25 years, with author Dr. Terry Flotte, UMass Chan Medical School, and From Concept to Cure, the Evolution of CAR T Cell Therapy, with author Dr. Carl June, University of Pennsylvania. Want to learn more about recent developments in the field directly from the people who are doing the research? There's still time to register and join us for ASGCT's 2025 annual meeting, May 13th to 17th, in New Orleans or virtually. If you become a member of the Society, you can save $465 on your registration for the premier event in the field. Check out the full program and register for the meeting by visiting annualmeeting.asgct.org. And now, Drs. Glorioso, Flotte, and June.
All right, well, I'm so happy you guys are here in one piece. And I'm looking forward to chatting with you about the amazing things that are going forward in gene therapy and cell therapy. And I looked at your reviews. They were just really fantastic and really informative. I mean, I thought Carl in particular did a great job in mapping out the history and so on. So it was really a great read. Really appreciate that, and people will love that when they get the journal. So Terry, how long have you been at Massachusetts Medical School, UMass Medical School?
18 years.
Wow.
Been dean and provost the whole time.
Wow. You look pretty good for that. That's a lot of politics.
But did I tell you I'm just 29 years old.
So Terry, do you want to describe what your interests are and how you got into this field and so on, and maybe talk a little bit about what you had planned?
So just to introduce myself, I'm Terry Flotte. I'm the professor and dean at UMass Chan Medical School, but also been a gene therapy scientist since long before that, really working from the beginning period of the creation of adeno-associated virus, or AAV, as a vector, kind of moving it from virus to vector. In the 80s, we had a role in that, and then really pushing it forward toward clinical applications, which we did at first at Barry Carter's lab at NIH, and then at Johns Hopkins, and then for 11 years at University of Florida in their gene therapy program. Let me just explain how I approached looking at the topic of gene therapy then and now. Yeah, so what we decided to do that might be a good way to get a snapshot of exactly what was going on in 2000 and compared to 2025 or over the quarter century after that, was that we did a systematic literature search to identify the trends, and we were particularly trying to make sure we could break down by different vector systems and different diseases and organs that were targeted. And I think this is really gives us the opportunity to talk about a few examples and how this sort of worked out. So a lot of gene therapy had kind of gained some steam by year 2000. And as it turns out, if you were to look at the vector systems that were in use, you'd see that of those that were being published in all the peer-reviewed literature, adenovirus had by far the predominant publication numbers in right around 2000 to 2004. And I think it's worth noting adenovirus has a very high rate of gene transfer in many different cell types. So one could easily get a lot of results, but it was certainly found through the Gelsinger case, the Jesse Gelsinger case, which happened in 99, and through many other systems where it was seen that the immunogenic and pro-inflammatory properties of adenovirus were limiting. So adenovirus really declined. Certainly has a role that persisted in cancer therapy and oncolytic therapy and in delivery of suicide genes in a kind of modified oncolytic framework. Also to say, gamma retrovirus vectors, those based on MMLV were, the Maloney-Murine leukemia virus, the original brand, they were still a big part of the gene therapy literature. AAV was just taking off, as was HSV, which, of course, Joe, I'm sure you're interested to see that. And lentivirus, I mean, the vectoring of lentivirus was newer. Vectoring, if you will, of HIV-1 to create the lentivirus backbones are used. Inderverma, Luigi Naldini, et cetera, had recently published that in the latter part of the 90s. And it seemed to really grow reciprocally with gamma retroviruses being less frequent in the literature. So we had a lot of discussion about adenovirus, and I would also say that this time was right at the end, I think, of a big push by the Cystic Fibrosis Foundation, which also helped to get AAV into the field. And I had the privilege of being the PI of the first clinical trial with AAV in a human, which was actually back in '95, which showed a much better safety profile than adenovirus. And some very different properties in terms of persistence. Non-viral vectors continued on, and it's interesting. Like at that point in time, you were only hearing about liposomes, maybe a few nanocarriers, nanoparticle carriers, but the current modality of choice, the LNPs, lipid nanoparticles, have come on very much recently and are now really having an enormous role in the delivery of CRISPR and many other non-viral activities. So, which organs was one using? Well, I think you can see a rise in CNS gene therapy that really paralleled the rise with AAV, and I think one of the things we found out in the AAV use in CF is that putting AAV into cells that are actively proliferating would result in the genomes being lost. And it became evident from that observation that the genomes were all episomal. And so it needed to be in non-dividing, terminally differentiated cells. And so the CNS applications arose along with AAV's rise. Certainly, the enormous unmet medical need of cancer remained there very much. And the other organ systems never really, as an organ system, really perked to the same level, but there's activity in all of those. So the diseases even more so to cancer, because some of the ones that were directed to hematopoietic stem cells or lymphocytes, of course, were cancer related, which you'll hear from Professor Jean very shortly. But neurologic disease, if you sort of plug it in by disease, and cardiovascular disease had kind of a heyday, but it's still such an enormous need. It stays high even as there aren't as many promising applications. And another one linked to AAV's rise is this shooting up here of eye diseases as the fourth most common disease type in the literature coming into 2025. Really interesting how the interplay between what the vectors we have will do and what things are applied to, how that plays out in terms of total activity across these different areas. And then, I did not in this talk bring the related to illustrative scientific publications, but I think there was a major breakthrough publication in 2009 that came out of Nationwide Children's Hospital. Brian Caspar and Kevin Faust, who I put a plug, was a graduate, got his PhD in my lab at Florida, was the lead author on that as a postdoc of seeing AAV9 cross the blood-brain barrier in newborn mice. And now that really led the way for a systemic AAV gene therapy to be used for neurologic disease. And then onto that led the amazing work Jerry Mendell did at Nationwide with spinal muscular atrophy type 1, which was really the real proof that you could get a dramatic cure in a life-threatening genetic disease. And so that's really laid the groundwork for where we are right now, where there's an awful lot of work in AAV gene therapy for severe neurologic genetic diseases. And in the article, we talked about a few other pivotal breakthrough examples, but I think that was just an interesting way to get at it. What were we doing back then? A lot of thought about CF, a lot of thought about adenovirus, and the conventional retroviral, gamma retroviral transduction of stem cells. And then in 2025, following along with what AAV works for, we have more brain and liver-directed gene therapy and eye-directed, and then seeing the amazing work with, instead of focusing exclusively on stem cells, hematopoietic stem cells, the modification of T cells, which you'll hear all about, to create CAR T cells, has revolutionized a whole subcategory of targeted immunotherapy. Which I will not kind of get into here.
Terry, excuse me, how many FDA approved products are there for AAV to treat rare genetic diseases? I guess most of them are rare genetic diseases, right?
Yeah. So I didn't update my list. I was up to 9 the last time I looked, I believe, of just the AAV ones. And some of them are duplicative. Oh, that's the total AAV approvals, I should say. So that includes the Luxturna, which is the eye for RPE65. That includes several hemophilia gene therapies for the liver, and that includes the FDA-approved Sarepta DMD drug, Elevidys. Yeah, so I think probably if you could just for severe neurologic diseases, it's still a small number, less than 5 in that range.
Yeah. Well, what's interesting though is that AAV has really been successful for treating rare genetic diseases, and probably there are many more. There are thousands of these, I guess.
Yes.
And the problem, I guess, is that there's a commercial issue, a commercial viability of treating these diseases where there are hundreds and now millions of patients.
Right, and that's kind of one of the points you make at the very end of that paper, which is that we have the tools potentially to treat many more of the 7 to 10,000 rare genetic diseases, but some of them that have been clinically successful are effectively market failures. So they hadn't been I hadn't really pictured this concept in prior years, but
I know, neither.
I know, yeah. We have some things that look really promising even in clinical data stage. Some of the AAV gene therapies for ultra-rare conditions that we're having trouble continuing the clinical program, and many others are as well, just because of the small market size and the way that biotech is now looking at that model. Certainly, very rare diseases have had a track record of at least having protein replacement, enzyme replacement therapy working, but that has a more of a conventional annual drug use. These one and done therapies where the dose costs $2 to $4 million. It's a difficult way to put together a business plan, because then you go years, you may treat an entire living cohort, and then the new cases are coming through at extremely low levels of new
Plus, many of these cases are in underdeveloped countries, right? So you have no way to pay for those. So the
Well,
democratization is another issue.
yeah, so as you know, I'm the incoming president starting in May of the HGCT, and we created a task force around access to gene therapy, as we were calling it. We have a principle for ASGCT of saying that we believe that there should be access to gene therapy for all who can benefit from it. And that really has two directions, right? One is the ultra rare, and we're really trying to embrace a very specific model for how we might help move that along. And more work will come along on that working group toward that. And then the other angle that a number of organizations are trying to get at is getting some of the gene therapies to where the diseases are most prevalent. So hemoglobinopathies are generally track where the equatorial areas where malaria exists. So that's true certainly of sickle cell disease and thalassemia to some extent. But the highest concentration for sickle cell, where we have two approved therapies, is in sub-Saharan Africa. And so there are a number of organizations that are addressing this. And I should say our task force is really composed of this coalition of all of these foundations that are doing this. But it's a little bit of a different problem for sickle cell gene therapy, because the gene therapy there is not just an in vivo administration of something you thaw in a vial and infuse. It really is in the context of an autologous hematopoietic stem cell transplant. And so, and while, it may be able to be done under less severe conditioning protocols than some others that are used for bone marrow transplant, it's not something one can assure can be delivered well or followed up well, or the patient's recovered well from the process, unless there's a substantial hospital with intensive care type resources, as opposed to the AAV, where in most cases it's a single infusion or injection, and the hospital infrastructure does not have to be as extensive.
Let me stop you there and give Carl a chance to enter the conversation. Again, your review was really enlightening, Carl. I really appreciate it. And maybe you could tell us a little bit about your very advanced program at Penn.
Well, thanks, Joe. And I think this is a nice bookend to what Terry just went over. So my comments really are on gene modified cell therapies. I began, clinical training in the early 1980s in bone marrow transplantation. So these were, in general, allogeneic sibling, HLA matched transplants. And that was really the first cell therapy for human tumors. And that was discovered by a really interesting finding when we did enough transplants in Seattle at the Fred Hutch that they had, most were sibling HLA match, but some were monozygotic twin donors. And the patients all got the same. They got high dose Cytoxan and total body irradiation. And so the only variable was the donor bone marrow and whether that was HLA matched or syngeneic as a monozygotic twin. And they did this because they thought there would be a lot less graft versus host disease with twins. But the surprise was that the anti-leukemia effect and the curative effect of the transplant was much higher if you had a brother sibling that matched or a sister than if you had an identical twin. So that was the first genetic proof in humans that there could be an anti-tumor effect. It really taught me a lot. I then studied along, about 20 years on how to grow T cells so that we could use autologous T cells that mediated this graft versus leukemia effect rather than, having to do allogeneic and a full-blown transplant. So in our paper that, it's really great to be able to write on the 25-year anniversary. because that's basically in 2000 is about when I started working on CAR T cells. So I initially spent about 20 years doing, T cell biology and working out how we could really optimize T cell growth and make robust systems to expand autologous T cells. But in our paper, we go through really the whole history of, immunotherapy, and it actually goes back to 18th century, with the first vaccines, with attenuated vaccines, things like cowpox.
Yeah.
And we learned a lot out of that. I mean, it showed that now, even today, that, if you have live viral vaccines or attenuated vaccines, you can get protection against not only the target, but also a more generalized trained immune system. And then Coley in the 19th century in New York started doing the first therapy for cancer using, killed bacteria. And that was Coley's toxin. And that never got FDA approved, but it did have some very striking responses. And that's where we learned about the innate immune system. We now know that basically those Coley's toxin were waking up macrophages, natural killer cells. And then, we had some dark ages where nothing much really happened from the Coley's toxins until we had the more modern technologies. We had to get the ability to transfer genes. And that happened in my case with gamma retroviruses and lentiviruses. So Dusty Miller and Michelle Sadelain, showed that you could modify T cells with gamma retroviruses. And then we did the first human gene transfer with lentiviruses. And it was actually an HIV initially to deliver payloads to patients with AIDS and modify their own T cells. And then,
Wow, that's
so that our first CAR T cell trial actually was in patients with AIDS. And we did this in the mid 90s with Cell Genesis. And Bruce Walker and Art Weiss had made a CAR that targeted the HIV envelope. And it had only the zeta chain signaling domain. So it was what we would now call a first generation CAR, and it could kill HIV infected cells. And we did a series of phase 1 and a phase 2 trial in AIDS and showed that it was safe and that there was some antiviral effect with autologous T cells expressing the CD4 zeta CAR in their CD8 T cells. So that was early data. And the amazing thing out of that is we found, because of the FDA's policy of following patients, for lifetime after they've been treated with
20 years or something.
15 years. And then now it's actually lifetime for CAR T cells. Lifetime follow-up is what the FDA is asking. But because of that, we had all of these patients that we treated with autologous CAR T cells in the 90s using a gamma retrovirus. We then, because if we could show that they no longer were engrafted with their CAR T cells, then we didn't have to follow them every year. So we did that in 2012. We took 40 patients we treated and analyzed their circulating cells, and we found that they still had CAR T cells more than a decade after we'd given them an infusion. So that was really the first demonstration you could permanently modify someone, with a gene modified cell and have long-term engraftment. And
Was they proliferate? These were acting like memory cells? Or?
Yes, yeah, exactly, Joe. They were memory CAR cells, and they were mostly CD8. And the most important thing was there's no side effects. No patients had either transformation or issues that we were worried about, like triggering autoimmunity, when we started amping up the immune system. So that we then had that in our sort of back pocket, that you could use genetically modified T cells and they could be safe for a long time. And then, about 3 academic groups were developing CAR cells. The first was made in the Weizmann Institute by Zeligeshwar, and that was a retargeted T cell that he made in petri dishes to show that you could use non-HLA and use antibody redirected T cells, which is the basis of a CAR recognition. But he never took that into trials. But then, my lab worked with CD19 CARs, making second generation ones that had a foreign BB signaling domain. And Rainier Brenchens and Michel Sadelain did this at Memorial Sloan Kettering with a CAR also targeting CD19, but having a CD28 signaling domain. And, we got grants from, the Alliance for Cancer Gene Therapy, both of us, in 2004. I was led by people,
Yes.
it was all philanthropy. There was no industry to do, cell and gene therapy because of something Terry mentioned, and there were a couple of high profile side effects in the late 90s. And then, really biotech decided and pharma that it wasn't possible to have a commercially successful cell and gene therapy. So they basically exited, and all of us were out in the wilderness doing, it was just academics. And so Michelle and I, Sadelain, and my group, both in 2010, had really striking responses with these second generation CARs in patients, with advanced leukemia. And then, biotech started coming back, and, Novartis licensed the CAR we made in 2013, which became Kymriah for, leukemia and lymphoma. And Kite licensed the one that Michel Sadelain had made, and that became Yescarta. And, so then now there's over 1,000 trials in the
1,000.
world. On clinicaltrials.gov, there's more than 1,000 CAR T cell trials. But most of them actually are now there's more actually numerically in China than in the US, which points to, I think, a large issue now of our system being able to rapidly develop, new cell and gene therapies, because it's now happening more rapidly in China.
Their funding is probably stable.
Yeah, well, there's funding issues, it's a national priority there. And biotech cell and gene therapy has not been a national priority in the US. I think this is something we really need to address. And then there's been the regulatory pathways for early stage trials are much more rapid, in ex-US than they are here in the US. So there's a lot of our trials that are either now and there's some of them are Australia, some of them are in China, rather than here in the US, which I think is a big issue. But so the first CAR was FDA approved that we made, in 2017. Now in 2025, there are 7 FDA approved CARs in the US. And there are projections with the cell and gene therapy. There's so many promising new indications being developed, and you can actually look and compare the rate of FDA approval. The previous, I think, disruptive therapy was recombinant protein technologies in the 90s that led to monoclonal antibodies like Rituxan, Herceptin. And there, in a 20-year period, only 10 of them were approved. And now it's 7 or 8 years since the first FDA approval. And, there are 7 CAR T cells approved. And then there's ones, that Terry mentions, there's gene modified stem cells, for, hemoglobinopathies. And there's now T cell receptor redirected T cells just approved this last year for sarcoma. So there's a huge pile up of, in these 1,000 trials that I mentioned of, many early stage trials. And then the issues are money and time of to get, trials completed, that until we have what I think is going to be hundreds of these cell and gene therapies, in the next decade. So it's a very exciting time. We have recently, right now we have these so-called second generation CARs that are the 7 FDA approved. They all have a co-stimulatory molecule. But this year there's big changes with so-called armored CARs. And a paper in Nature recently in a solid tumor from Baylor showed really potent effects in neuroblastoma with a CAR targeting GD2 and having a IL-15 expressed in the CAR cells. And then we have exciting data now in patients who've relapsed after CAR therapy with a commercially approved CARs. And now we've armored the CAR T cells. They're CD19 specific still, but they have the IL-18 cytokine, which is secreted, and that activates natural killer cells in the patients, and it really improves the potency. So there's a lot of next generation approaches happening. And over the last year, I think there's been two major changes that we're going to see. One is 3 groups reported, phase 1 trials. This was City of Hope, and the Mass General group led by Marcella Moss, and then our group led by Don O'Rourke, where we have CAR T cells for glioblastoma. And, this is our 3rd trial now, and we now have 18 patients treated and quite striking responses. And that's true at the other institutions. So in our case, we have a dual targeted CAR, and, for the glioblastoma.
IL-13 receptor and EGFR or something like that.
That's exactly right, Joe. So there's, because we initially did trials just targeting EGFR, actually the mutated EGFR variant 3, and we had target escape. So the tumor would come back and it would lose a target. And now with dual targeting, as you mentioned, IL-13 receptor alpha 2 and, mutated anti-EGFR CAR, we're seeing much less target escape. So, we're learning from the early trials that didn't work. And I think that's really exciting to see, because there's all of the blood cancers, there's really striking data in clinical trials, but solid tumors, the results until just recently have been disappointing. The other major thing we're seeing is, and this came out of academic trials in Germany, in Erlangen, is amazing results with CD19 and BCMA CARs. They're used normally for myeloma.
Myeloma, yeah.
Many groups now have seen very striking results in awful systemic autoimmune diseases like lupus and scleroderma and inflammatory myositis. And there's now over 80 trials, when I looked up on clinicaltrials.gov, of CAR T cell trials for autoimmunity.
That's a fantastic next step.
So it turns out basically the same kind of CAR design that's been optimized for cancer therapy has a very striking effect in people with autoimmunity. And that was really unexpected, because all the mouse models really for autoimmunity, people thought that it was due to autoreactive T cells, and that you had to immunosuppress the patient, with chronic corticosteroids and TNF blockade, et cetera. And now what the data really from the group in Germany shows is that it's this idea that it's a so-called an immune reset, or really just a rebooting of the immune system. They just have to give a CAR T cells and have transient B cell aplasia. And then, the pathogenic antibodies, like against double-stranded DNA that you see in lupus, they just go away and they haven't come back.
Wow.
It's been, no one thought that would happen based on what was done in mice. So it points out sometimes that, our models aren't adequate. We've really had a history with
Humans are better than mice.
Yeah. But as you all know, and I think all of us experience, the animal models have not been good really in predicting toxicity.
Right.
And you have to do human trials, but so anyway, it's a very exciting thing that we're going to see CAR T cells for autoimmunity. And I'm pretty sure, there's, lots of groups have shown that cell therapies, both with stem cell therapies, like has been done out the Fred Hutch by Hans Peter Kim over the years, and T cell therapies can actually make cures in HIV. The problem is that it's not economically, the cost is too much compared to taking antiretroviral medicines every day. But I think, these cell and gene therapies will have potent effects on, chronic infections, as well as now autoimmunity and cancer.
I guess at this meeting that you and I attended in New York, I think that there was a lot of interest in vivo CAR T production. What's your take on that? Are you guys doing similar experiments in PET?
On what kind, Joe?
some kind of in vivo
Oh yeah.
development
So
to create CAR T cells in situ.
yeah, and this is an amazing area as well. So now both there are several companies, biotechs giving using viral vectors, either retargeted lentiviruses that can get into T cells. For instance, instead of VSBG, they may get in via an anti-CD7 or CD8,
Hmm.
and then infect cells in vivo, and then the CAR T cells have been made in the patients with no ex vivo manufacturing.
Hmm.
And so that is, as and early results look really promising from those trials. Both AAV has been used that way, and then now viral-like particles. So in vivo manufacturing of the CAR T cells look promising. And then with Drew Weissman, our group here at the University of Pennsylvania showed we could use antibody retargeted lipid nanoparticles
Right.
to deliver RNA cargo and make either CAR T cells in vivo, and it's really amazing in its efficacy. We could get about 80% of all the T cells in a mouse become a CAR T cell
Wow.
by injecting in the tail vein these lipid
Wow.
nanoparticles, and so and we showed that could have effects in both cancer and also in like cardiac regeneration in mice that had cardiac injury. So we're really excited that these in vivo technologies will be there's much more scalable. It brings out the issue Terry was discussing about being able to, for instance, deliver something like this in sub-Saharan Africa where there's no cold chain, and it would be
Right.
possible to do an injection like this, but really to do, a cell therapy with gene modified stem cells is just not going to be possible to scale, in that setting.
Would these require a kill switch, you think? So that
Well, I think so. I mean, I think we all know where there's been issues with insertional oncogenesis with viral vectors and recombination and so on. And so with in vivo injection of live viruses, there's also the potential of germline transmission.
oh
And
yeah.
so, that is something that's going to be very carefully watched with live viruses being used in vivo. But I think we don't have first generation viral vectors anymore. I mean, I think all of us of this age on this podcast know what happened, in the 80s when recombinant retroviruses were injected in non-human primates, and that it resulted in T cell lymphomas.
Right. Right.
Because but now we have the SIN vectors. The technology is much better to prevent RCLs and recombinant RCRs. So, but I think it's too early to know that it's going to be safe to inject, an integrating virus in people in vivo. But to me, it's even amazing that it can even be done.
Yeah.
If
I,
I could ask a quick question, I don't mean that if that's the out of order for the podcast, but just how much of this is now being addressed with editing, like editing into the gene editing into the track locus, et cetera, to try to do something a little different than random site integration?
Yeah, so that's a great question, Terry. I know that at UCSF, this is being done by one of Michelle Sadeline's trainees, Justin Ekim. So he's using AAVs and then a combination with a viral-like particle to deliver guide RNAs and then knock it into the track locus, exactly as you suggested. And I think that will make safe harbor integration sites should make this much safer. But it's also possible these, RNA payloads with lipid nanoparticles where you could knock into locus with and have like a Cre-Lock system. So you could have nuclease system. It's really going to be an amazing new frontier, I think.
That's very exciting. So I guess like with AV and gene therapy, CAR Ts currently have the complication of being manufacturing issues. It's very expensive and difficult, and very few people have access to it. And so the in vivo approach seems like that could be the next generation, but I can imagine that these patients had to be carefully monitored for development of tumors and so on.
Yeah, that's exactly right. I think there's going to be both the ex vivo cell engineering, and that's going to be two flavors. It's going to be either autologous, and because we know those can survive for more than a decade in patients. We've now seen that in our first patients treated that had leukemia back in 2010. They're still engrafted with CAR cells, and but also we're going to see, so-called off-the-shelf sources. They may be derived from cord blood, umbilical cord blood, and where there are trials using natural killer cells that are gene modified, or and then those can be given, and they don't cause graft versus host disease, so they're
Right.
autologous, and they're cheaper to manufacture. So there's a number of trials with the so-called allogeneic engineered cell therapies, but I think there will always be a spot for ex vivo, because for solid tumors, what the field has found is that you need to do a lot of genetic editing on top of retargeting the cells. So
Right.
if you're knocking out 4 different genes at once, for instance, and adding in a transgene like a cytokine, that's going to be very difficult to do in vivo.
Exactly. Yeah.
But it may be sufficient, I think, just to hit one gene, I think for in vivo for autoimmunity,
Mhm.
and some, more simple cancers, but not the highly. I mean, some of the solid tumors have so many different mutations, and I think that's going to be best treated with ex vivo manufactured cells. But a lot of change is going to happen there. It's becoming automated instead of done by, highly trained technicians and scientists. And then, so that's work in the industry that I think is going to really lower the cost of goods. And then there'll be, point of service and point of care, probably turnkey boxes made where, just kind of like we do our Red Cross blood platelet donations locally at our own healthcare centers. I think that'll happen with manufacturing of, both probably stem cells, natural killer cells, and T cells at point of service.
That's fantastic. One of the things that for both of these that are kind of an issue is safety, and I think that both for AAV as well as some CAR T experiments, there are adverse events, and Terry had some experience with a recent one that with an AAV driving a mini gene for dystrophin. And one of the problems I think would be that how do we prep these patients so that they're more resistant to these adverse events? If you introduce into a solid tumor vectors that don't kill tumor cells, but rather express cytokines that might recruit CAR T cells, do you think that would allow better entry into the solid tumors?
Oh yeah, for sure. We have data on that actually now. First that was done at UNC by Jim Petrodoti. He showed that he used an adenoviral vector, and that, quote, heated up the tumor microenvironment, and then it was in a glioma model in mice, and then he got much better recruitment of CAR T cells into the tumor, which, at baseline was very cold. There was no lymphocytes there.
Right.
So I think there's a lot of synergies going to happen with combining viral vectors and cell therapies. So that is for sure in the, preclinical models. It's difficult in practice to get these trials open on a regulatory thing, because you're combining,
Right.
a viral vector, the people who
A
review
lot of
that.
manufacturing issues. Yeah.
Yeah, it's a hard thing to get, but the data is really compelling in mice.
I mean, one of the most difficult tumors, I guess, besides GBM, is pancreatic cancer.
Yeah.
It's a very difficult tumor because it's so firm and it's difficult to get things in. Are you guys considering any novel approaches to treating pancreatic cancer?
Well, we done two things there, Joe. One, because you know that it's so one thing is a tumor can suppress T cells or kill them. And
Mhm.
then another is there can be physical barriers, like you said. And then in pancreatic cancer, there are these inflammatory fibroblasts which lay down collagen, and it's basically a scar. It's like a castle surrounded by a moat.
Yes.
The cells can't get in.
Yes.
And it also impedes, like if you give checkpoint antibodies
Right.
or viral vectors, they just don't get in. And so we had an approach last year that we published with Ellen Pure here at the Wistar Institute. And what we did was give two different CAR T cells. So one was targeting the fibroblast, and we targeted the molecule fibroblast activation protein.
Oh, that's cool.
So
Yeah.
it's a FAP CAR is what we call it.
Fat car.
And we did this in the so-called KPC mouse, which they have, tumor. It's a KRAS-driven tumor in the pancreas. So and if we gave a CAR targeting the cancer cells, which was mesothelin, nothing happened. Zero. There were butkus. There were no T cells that got in the tumor. But if Ellen gave the anti-FAP CARs and then waited a week, and then gave anti-mesothelin CAR cells that targeted the cancer cells, we could cure mice.
Wow.
So in one
Great.
week, you can get a lot of remodeling, at least in this mouse model,
Mhm.
of this impenetrable barrier. And she showed that, looking at, from sophisticated microscopy, that the collagen barriers were broken down when she killed the fibroblasts.
Fantastic. Fantastic. Well, guys, we're going to have to probably wrap this up. But I have to say that it's been a very exciting set of conversations from both of you guys. And it's such a pleasure to talk to principal leaders in the field of cell and gene therapy. And I'm so happy that you're on the case, and I'm sure there's gonna be many exciting breakthroughs in the future. So again, I look forward to seeing you in New Orleans, I hope.
Yep.
And so that'll be a good time we can catch up more.
Thanks, Terry. Thanks, Joe.
Yeah,
Okay.
indeed. We'll
Yep.
see you in New Orleans.
Yeah. Thank you.